Hello everyone, very good morning. Welcome to Knowledge Hub by Sri Chaitanya. I am Pratyusha, Senior Botany Faculty, and in this session we are going to do the continuation from our last session. That is the remaining two classes of Kingdom Fungi. So in our last session we have studied in details regarding the Phycomycetes and Ascomycetes from the Kingdom Fungi. And in this session we are going to do the next two classes, that is Basidiomycetes and Deuteromycetes under the kingdom fungi. So let us start the session, and I hope this will be really very helpful for your upcoming NEET exam. And do not forget to like, share and subscribe, and stay tuned with us till the end of the session to get a better grasp on this concept, right? So let us begin. Okay, so the first thing here is that regarding the first group, that is Basidiomycetes, they have a common name. Apart from being called as just simply Basidiomycetes, they are also called as something called as club fungi. Now, why does this name club fungi come? Because it has been observed that they are clubbing together. Okay, so clubbing together give them a feature properly. So as you can see that, like in a group of mushrooms, they will be placed like this. Okay, all of them will be like somehow like this. So you can see that the group of mushrooms, they are basically clubbing together, right? Hence they are called as the club fungi. Now let us talk about their habitat. So in habitat, you can see they are present here also. This one is also more of the cosmopolitan, right? So if I say cosmopolitan, then what is here? So they are present in the soil, they are also present in the logs and also in the tree stumps. Okay, anywhere in the tree, okay, present in the tree, in the blocks of the trees, in all those places. They are present in the tree stumps, they are present in the logs, and also they are present in the living plant bodies as parasites. So you can get one more feature from here, that they are free living. I mean, by free living they can be saprophytic, and if they are present in some of the plant bodies, then they will be parasitic, right? So now let's see the other properties of the Basidiomycetes. Now, next thing is the distinction of the mycelium, right? So mycelium you have studied in the general characteristics of the kingdom fungi, that the mycelium is branched and septate for Basidiomycetes, right? This is the characteristic of the Basidiomycetes. So branched, I mean it will be something like this. Okay, if the mycelium is there, I mean which is making up the entire, it will be branched, and there can be multiple branchings present, right? And septate means it will be something like this. Okay, here it can be having many of this. Septa is this wall. So this is number one. This one is the branching. Okay, this is the branching, right? I hope you all can see this properly. This is the branching, and this is the partition, or you can call it the septa, or it is septate, right? Septate means it is having partition, proper partition between the two nucleus. Okay, so this is partition, or you can also call it septa. Okay, now what is the nature of the septa? This is a bit more extension to your NCRT, right? So in your NCRT you have that Basidiomycetes, they are septate and they are branched. In addition to NCRT, how do the septa look like? Okay, so they are having dolipod septa. New word, what is dolipod? Okay, let me explain you what is dolipod septa. I hope I can erase this because you all have got what is this branching and also what is this partitioning, that is the septa, right? So branching you have got, and the partitioning also you have got, that is called as the septa here, right? Now let us see here once. So branching and the partitioning, everything is done. Okay. So now let's see here, dolypore septa. What do you mean by dolypore septa? We have two types of septa. Simple septa, which is just formed due to the coming together of both the different. Let's see, this is a hyphae. Okay, this is hyphae number 1 and this is number 2. Okay, now in between them there will be a septate, right? Or there will be like septa. Okay, this is the partitioning formed. Now this is also one septa, this is also one septa. Both of them will come together, they'll have this kind of structure which is joining. Okay, I mean if it is like this, okay, so one will be coming from here, one will be coming from here, here they will join, and there will be a small pole like structure for transferring something. This is what, this is for transferring, but this is both are closed. Okay, they're not allowing anything to pass or like that. This is completely closed, but this is formed due to the coming of the two. Let's say this is the nucleus, here it is there, and both have started together. Okay, from here also one, from here also one, right? They'll form the proper septa. Okay, so in order to form this, it will start from here and it will start from here, and both of them will come together, and this one is very much proper as a dolypore septa. It will start from the both opposite sides. Okay, then they'll come together, they'll form a ring like structure, like this kind of this. Okay, so this is dolypore septa, and we have various names for these things. Number 1, first thing is this, the upper structure of this, okay, which is surrounding the dolypore septa, okay, which is surrounding the dolypore, in compassing this dolypore from the upper side, that is called as a pore cap. Okay, that is called as a pore cap. Pore cap is also called as, one more thing, it is also called as parenthysome. Okay, so this is another name of the pore cap. I hope you will just remember the pore cap, or better, it's not necessary for you to completely remember the structure of the dolypore septa. You can just remember how the dolypore septa is formed. That's it for your NEET. Okay, or you can just remember that the dolypore septa is present in the Basidiomycetes. Right. Now, so dolypore septa is present in the Basidiomycetes. We have one more feature after this. What is that? That is the primary mycelium. Okay, so primary mycelium is the mycelium which comes first. Right inside this, it will come. Okay, so you know mycelium is what? It is making the inner structure of this. Okay, so it's making the inner structure of this. This is the mycelium. Okay, this is the mycelium. Mycelium is comprising the inner structure. Now here, so after this, what else we have is that, so mycelium is comprising the inner structure. Okay, let's see, there is one single first mycelium which is formed first. Okay, mycelium which is formed first. From here, another, it will give rise to another type of mycelium. Okay, not this one, but another one. Okay, this will go away. It will be replaced with another type of mycelium which is long lived and extensive. Like how we humans have a primary, I mean, we have two types of teeth, right? Milk teeth and permanent teeth. It is something like this. First, the entire body will be having the primary mycelium. So after the primary mycelium, after few days, it will be replaced by the secondary mycelium, which is long lived and it is more extensive. Extensive in what sense? Extensive in branching. Okay, so it is more extensive in branching, right? Okay, now let's move to the next thing. What else we have? We have three types of reproduction. Again, all the types of reproduction are observed very nicely in the Basidiomycetes. Okay, so here we can see asexual spores, they are not generally formed. What is the more important type of reproduction? Through vegetative fragmentation. Okay, vegetative reproduction is more common. Okay, so asexual spores are not present. Okay, one more thing, asexual spores are not present, it doesn't mean that asexual reproduction is completely absent. No, asexual reproduction is present very much. We can see here vegetative propagation, it's formed, it's a type of asexual reproduction, you can say that, but asexual spores are not present. Next thing is that sex organs are absent, but sexual spores are present. Okay, so sexual spores are present, but the sex organs are absent, right? So now we can see, we can go into a bit details about how the reproduction happens in Basidiomycetes. Let us look at this. Let us see one hyphae. Okay, so this is one hyphae from a mycelium, and this is another hyphae from a mycelium. What is the difference? Why one is marked with plus and the other one is marked with minus? Because this will be the donor and this will be the receiver. Okay, so you can call this donor for some time and you can call this recipient. Okay, so now what will happen here, you can see this will be fusing. So this will be taking this one, it will be taking the nucleus of this one, it will be taking the nucleus of this one. Okay, let them fuse. So they have fused, but everything has fused except what? The nuclei did not fuse. So nuclei has not fused. We can see what has happened here, plasmogamy has happened. Okay, there is fusion of the cell wall, so both the walls has fused, right, properly. So we have this two nuclei which still has not fused. So this is still undergoing fusion. We can see first compartment, okay, this is divided by septa, all of this. So first compartment has undergone fusion of the cell wall. Okay, so chitinous cell wall that has undergone fusion. First one, we have this. Next one also, we have this. Okay, it has fused except the nuclei. Everything has fused except the nuclei. Next thing, so finally this one also fuses. Okay, it stays for some time in this stage. What is this stage? This one is having double nuclei. Okay, this is having two nuclei, but the cell is fused, right? So this is an example of plasmogamy which is brought about by the fusion of two vegetative or somatic cells of different strains or genotypes. Okay, so both of this donor, this recipient, this both are of the different strains. Okay, so now what will happen after this? We can see here in each of the compartment, okay, in each of the septa, in each of this division, it is having two nuclei, it is not having one. So the nuclear fusion is not there, but the cell wall fusion is there. So here, do you think nuclear fusion will happen or not? Yes or no? Please hit the comment section. I will explain you what is this. Yes, my dear students, if someone has answered yes, you are very much correct. I can tell you karyogamy will happen. But this will take some time. Okay, it will take some time to come into the karyogamy. Once the plasmogamy has happened, after that, after some time, okay, after some time, the karyogamy will happen for sure. Okay, then this is N and this is also N. Okay, now this will become, this is N plus N. Okay, and each of these compartments are having N plus N, right? So each of these compartments are having N plus N. So after this, there will be, okay. So now we can see this one more thing. So we have different types of mushrooms. So one of the mushrooms, they are button mushroom, and if it is coming to the elder form, then we can call it sprout. Okay, it has sprouted up. So completely, initially it will be button. After the fusion of this, okay, when this karyogamy is still pending, at that time this will be button, in a button state. It will be just, it's just looking like a pop-up, right? So it is having the shape of that of a button. Now after this will take its original umbrella shape. So that time we will call it a sprout. Okay, So, okay, now let's see the next thing, that is further reproduction further. What will happen after this? So this has sprouted, okay. Now let us come to the different part labeling of the sprout, right? So what do they have? They have stipe, that is the holding thing, okay. This is basically like that of a stem of a plant, okay. This is like a stem of a plant. You can see, you can compare the structure with the umbrella, okay. So umbrella is having a handle. So handle is similar to the stipe, okay, which is holding it, and the umbrella is having iron spokes, right? So umbrella is having iron spokes. So this mushroom is also having what you call this gills, okay. So this gills, or this part, particularly this part, okay. So this part is there, which is providing the spokes, the spokey structure to this. So this is the gills, this part, right? So then this part, iron spokes, okay, is similar to the iron spokes. Finally, they have the cap, okay. So they have the cap of the umbrella, which is called as a pileus, okay. It's called as a pileus for the mushroom, right? So explain the structure of the mushroom very clearly, similar to that of an umbrella. Each structure is similar. The handle, stipe, let me repeat, and the iron spokes, gills, and the cap of the umbrella is the pileus, right? So let's, all those who can't understand it properly, let's just write it once more. Okay. Okay, now we can see the further inner structure of the gill. Okay, if you take a cross section of this, okay, let's just take this color. You take a cross section of the gill and you look at the cross section of the gill here. So then what you will have, the gills are having much more inner peculiarities, okay. So the inner peculiarities of the gill, we can see that this is having a basidium, okay, the outer side, okay, and attached to the basidium there are this small structures, they are called the basidiospores. So these are, my dear students, the sexual spores of the Basidiomycetes. So what we have learned just like this few minutes back, asexual spores are generally not found, right? And sex organs are absent, so they definitely have the sexual spores. And the sexual spores here is the basidiospores. Okay, basidiospores are the sexual spores present in the Basidiomycetes. They are present in the outer part of the basidium. Okay, now this part, which is comprising of the basidium of the paraphysis, is totally called as the hymenium. Hymenium is exactly the outer part of it. Complete outer part is called as the hymenium. It's like the hymen part of it, okay. Next, inner part, some circular inner structures, okay, coiled inner structures, they are called as the trama, TRMA, okay. And even further coiled structures inside of it, that is subimenium. Okay, so all this, it's not that much important. You can just see the inner structure of the gills. I am telling you, which is very important, that is the basidiospores, okay. So basidiospores, how it is located? Towards the outer side like this, and exactly outer side of the gill. So this is agaricus's internal structure. Agaricus is what? Agaricus is a mushroom, right? So common name is the button mushroom, or whatever mushroom we call it. Button mushroom, stout mushroom, all these things are there for the agaricus, and agaricus's internal structure of the gill is like this, right? Okay, now let's see further. Okay, so inside this you can see, so you have got the idea of this, what is happening here. So in this particular structure, that is the hymenium, or we can say this particular structure, that is a basidium. Okay, in the basidium of the hymenium, inside the basidium, finally what will happen? The karyogamy will take place. Okay, both of them will unite. Okay, both of them will unite, both the nucleuses will unite and they will form a double nucleus. Okay, the double nucleus will be 2n. That will be present in the basidium. Further, immediately after, sorry, this is a bit, just needs a bit change. So let's imagine the fusion has taken place here. Okay, let's imagine the fusion has taken place here itself and now it has become 2n. Okay, immediately after it has become 2n, what will happen? Immediately after it has become 2n, it will undergo meiosis. Okay, it will undergo meiosis and it will form the basidiospores. Okay, two different types of spores because it came from the two different parents, plus and minus. Remember? So now, plus and minus, two different spores. So you can distinguish this as male, female, something like that, but it's okay. It depends on the strains. Okay, now the spores will be generated. It will be liberated. After the generation of the spores, outer side of this will go up, this will form the spores. You can see the spores present here, and from here what will happen? This will come up, okay, this will fall on the grounds, this will undergo germination in the presence of favorable conditions. Okay, so if favorable conditions are present, what are the favorable conditions? Proper temperature, proper moisture. Okay, and see, mushrooms, all of them, like they like staying in warm, shady and moist places. Okay, so too much cold is also not very good for them, not favorable for them, and too much sunny areas are also not favorable for the mushrooms. Okay, after this has come out, this will land, this will form the primary mycelium. Okay, this will form the primary mycelium. And after this, what will happen? From here, you can see that the basidium, this will undergo karyogamy. Okay, so basidium has undergone karyogamy, it has formed the basidiospores. So basidiospores will, after this, continuation from here. Just remember, basidiospores, we were here. So from here, it will be forming the primary mycelium. Okay, from the primary mycelium, this will undergo again plasmogamy. Okay, so these are what? Just single in each. Okay, so two of them will fuse. Okay, so second, it will form the secondary mycelium. Okay, secondary mycelium, this is made up of dikaryotic cells. So it's just the fusion of these two. It will form the secondary mycelium. Fine. From the secondary mycelium, it will further undergo formation of this first button stage. Button stage will enlarge. It will form the fruiting bodies, that is the aerial part of the fruiting. This fruiting body, this part, this is called as the carp. Okay, since this is Basidiomycetes, we are going to call it Basidiocarp. Okay, Basidiocarp for the Basidiomycetes. Now let's see this one. Let's see some of the examples of the Basidiomycetes. So these two examples which is given here, these two are parasitic. Previously you had the life cycle of Puccinia graminis tritici. So this is a wheat parasite. Typically this is a wheat parasite. Okay, so all of you please do remember the scientific name of this Puccinia graminis tritici. Puccinia is causing the wheat rust. Okay, disease is the wheat rust. You can call it rust or you can call it wheat rust, you can call it both. Okay, so this is a parasite for the wheat and this causes the wheat rust. Let's see the other one, Eustilago. Eustilago, this one is forming as another disease. This is also another parasitic. Okay, so this is also parasitic and this causes smut in the maize. S-M-U-T, smut. So smut in the maize, okay, this causes, fine. So this causes smut in the maize, and Puccinia, it causes rust in the wheat. So now, next thing is agaricus or mushroom. So mushroom or agaricus, these are safe, okay. So this can be eaten, this is edible, mushroom is edible, but my dear students, amanita or toadstool, this is a poisonous, okay. This is poisonous fungi, okay, and this is mushroom is generally edible except few, okay. So I'm just mentioning this is generally edible, but both of them are not parasitic, they are saprophytic in nature. Let's see the other examples, we have others, okay, of this also. So polyporus, this is called as a bracket fungi, it's all of this are given in NCERT. So this examples is very important and it matters, and please remember the pictures, okay. So please do remember the pictures, whatever picture is given in NCERT, you have to remember that, right. Picture related question can also come in NEET, okay, my dear students. Next thing is lycoperdon. So lycoperdon or puffballs is also very important, right. Let's see the other one. So now, see, this is a comparison between Ascomycetes and Basidiomycetes. If you want to write it down, you can pause this video here and you can note it down, okay. I'll just move step aside for 1 minute. If you want to write down any one, you can just write for this entire thing, this entire chart. It will be very helpful for your NEET exam. So I'm still repeating, you can write this down. You can give this is a proper comparison between the Ascomycetes and the Basidiomycetes. Both of them are very important classes of fungi for your NEET exam. Okay, let's see the Ascomycetes first. So what happens in case of Ascomycetes? We have the predominant primary mycelium. So primary mycelium, I think all of you have remembered, this is like this, right? Now, what for the Basidiomycetes? For the Basidiomycetes, the primary mycelium is short-lived and it gets replaced by the secondary mycelium. Secondary mycelium will be something like this, okay. So here, this is formed due to not forming of the karyogamy, okay. So from primary mycelium, you have this as a secondary mycelium, okay. Now, secondary mycelium in case of Ascomycetes is short-lived, and secondary mycelium in case of Basidiomycetes is long-lived, okay. It is much longer living. Next thing is asexual spores. So asexual spores is absent for the Basidiomycetes. It's not present, but for the Ascomycetes, we have very nice asexual spores, which is what? Exogenous or endogenous? Please write in the comment section. I want this answer, right? Or if you want me to answer, want me to teach you, please come to our nearest center. Okay, so conidia is what? What, my dear students? Exogenous spores or endogenous spores? You will get the answer if you ask the question in the comment section. Okay, so I'll not answer here. I'll answer if you drop a comment, right? Okay. Now let's see for this site for karyogamy and meiosis is what? Site for karyogamy and meiosis is the ascus, okay. But for the Basidiomycetes, it is the basidium, okay. So site for karyogamy and meiosis is the basidium or the Basidiomycetes. And now let's come to the fruiting body. So fruiting body is called as the ascocarp, but for the Basidiomycetes, it is called as a basidiocarp. And the last one, last but not the least, here we have the exogenous basidiospores, but for Ascomycetes, we have the endogenous ascospores, okay. So many of you might have got the answer, okay. If still not, please come and let's get started with the lessons, right? Okay, let's come to the next one. So we are talking about now the new group. That is the last group, that is the Deuteromycetes. Okay, Deuteromycetes, let's come to the characteristics of Deuteromycetes, how it is like. So mycelium for the Deuteromycetes, this is septate and it is branched and it is colorless. It is not having any color. So you can see the innermost part also, okay, how the branching and how the septation and how everything is taking place, right? So now, one more thing, my dear students, nutrition. So in the nutrition way, this is saprophytes, or you can also call it decomposers. So now, my dear students, a very, very, very important line from the NCERT is this. Okay, so this is what Deuteromycetes helps in mineral cycling. So many of the processes, formation of petroleum, formation of many, I mean like all these things, minerals and all, okay, this depends on one important class of the fungi, that is the Deuteromycetes. Okay, it's also called as fungi imperfecti. Why? I'll just come in the next slide. Okay, and next also they are parasitic. Okay, so I told you fungi are having two important classes, whether it is saprophytic or it is parasitic. So this one is parasitic. Okay, so now saprophytic, parasitic both are present for the Deuteromycetes. Let's see the next class, next group. Okay, so let's see here. Asexual reproduction is through conidia for the Deuteromycetes, but here sexual reproduction or sexual forms of the Deuteromycetes is completely absent. Yes, absent. Hence, it is called as a fungi imperfecti. Okay. So this is called as a fungi imperfecti because it's not having the sexual form. It's not having the sexual stage. If you discover the sexual stage, if any fungi which is belonging to the Deuteromycetes group, let it be Fusarium moniliform, okay, moniliform or Gibberella fujicuroi, both of them. Okay, so this is, once it was found out, so this has moved from Deuteromycetes to Ascomycetes. Okay, this one is now called as Gibberella fujicuroi. Okay, this has moved from Deuteromycetes to Ascomycetes. There can also be moving from the Deuteromycetes to Basidiomycetes if the important feature of, that is the sexual stage of the fungi belonging to the Deuteromycetes, can be discovered. Okay, if they can be discovered, then they will be moved into the classes where they rightly belong to. That is these two classes, Asco and Basidio. So once it can happen that all the different fungi belonging to Deuteromycetes might be having their stages of sexual form discovered, and then they'll move to both of these groups. Okay, because Deuteromycetes have all the properties, maximum properties are similar to Ascomycetes and Basidiomycetes, right? They are not at all similar to the Phycomycetes. It's completely different. If you see the evolutionary changes, evolutionary development, okay, right? So Phycomycetes is completely separate, and then you have the rest of the three groups, Asco, Basidio, Deutero. All of them are more or less a bit similar, not I'm saying that much similar, but a bit similarity is present, right? I hope you got this, all of you have got this very well, and you have prepared very nicely for your NEET exam by this chapter, by this lesson, right? So let's see some of the more characteristics of the Deuteromycetes. You can see what is this? This is causing few of the important diseases. Okay, here. Okay, so after this, let's take some of the few NEET questions. Okay, these are just the what diseases is caused by the Deuteromycetes group, and let's take few questions which will give you a thorough revision of the entire kingdom fungi. Okay, so ready? Let's begin. First question here on the screen, which says which of the following statements is incorrect. So you have to guess the incorrect statement. You just have 5 seconds for it. Okay, it's a very easy question, I think, from both the groups, okay, from all the fungi groups. So let's see the first group. Yeasts have filamentous bodies with long thread like hyphae. Is it true? Yeasts are belonging to which class? Okay. Next thing is morels and truffles. They are edible delicacies. Okay. Now, next one. Claviceps purpurea. This is a source of many alkaloids and LSD. Right? Is it true? Okay, now let's see the other one. Conidia are produced exogenously and ascospores are produced endogenously. Okay, how conidia is different from ascospores? Let us look at the answer. I hope you all have got enough time. Let's see the answer. The answer is the first one. Okay, first one is absolutely incorrect. Okay, which says that yeasts have filamentous body with long thread-like hyphae. They don't have. They are the exceptions to this. Okay, so yeasts are the unicellular ones. They have many other characteristic features similar to that of the fungi, but they don't have this filamentous body thing. Okay, so now let's see this. After karyogamy followed by meiosis. Okay, karyogamy, then meiosis. Spores are produced exogenously in which group? You have to look at the groups. Okay, you have to see the proper names of the fungi which is belonging to the different groups, and you have to answer the question. First is Neurospora. Okay, next one is Alternaria. After that, it is the Agaricus, and then we have the Saccharomyces. Okay, so few are belonging to few different groups: Phycomycetes, Ascomycetes, Basidiomycetes, all these groups. So this is the characteristic feature of which group? Okay, this is the feature of which group of fungi? You have to look and you have to answer. Ready for the answer? I hope you all are, right? So the answer is Agaricus. Agaricus is rightly belonging to Basidiomycetes group. Okay, so Basidiomycetes group, this is having this important characteristic of the karyogamy. What is this? So after karyogamy, okay, so karyogamy, first of all, it is delayed. Okay, it is a prolonged karyogamy which we are having in the Agaricus. So in Agaricus, we are first of all having a prolonged karyogamy. After the prolonged karyogamy, it is finally having meiosis. Okay, so after meiosis, the spores are produced, and the spores are produced exogenously, right? So this is the answer. So answer is Agaricus, which is the mushroom, okay, belonging to Basidiomycetes. Now, let's see the other question fast. So, cell wall is present in the members of fungi and plantae. So, we have to do what? We have to select the wrong statement out of this four. Okay. So, cell wall is present in the members of fungi and plantae. Is it true or false? If it is false, you have to mark this statement. Okay. Next thing is mushrooms belong to Basidiomycetes. Okay, after this, pseudopodia. Pseudopodia are locomotory and they are the feeding structure in the sporozoans. Okay, so pseudopodia, this is a question from the kingdom Protista. Okay, so pseudopodia are locomotory and feeding structure in sporozoans. Is it true or false? Next is mitochondria are the powerhouse of the cell in all kingdoms except Monera. Now, let's look at the answer. So, obviously the third one is incorrect. You can look at this. Sporozoans do not have any locomotory organelle. Sporozoans lack any sort of locomotory organelle, including the pseudopodia. Pseudopodia is present in the Sarcodina group, not the sporozoans, right? Now, all these are true. Cell wall is definitely present in the members of fungi and also in the members of plantae. So, fungi is having cell wall made up of chitin and plantae is having cell wall made up of cellulose. Next thing is mushrooms. They definitely are the Agaricus scientific name and they belong to the Basidiomycetes group. Last one, mitochondria is yes present in all the groups, but it is not present in the monadans, right? Okay, let's see the next question. Another very interesting one. So this is a direct question from the fungi, okay, without any other groups, without any other intervention of the groups. So let's see the imperfect fungi, which are the decomposers of the litter and help in the mineral cycling. We have just read this one, right? So you have to answer this question, my dear students. So this is present in which group? It's a characteristic of Phycomycetes, Ascomycetes, Deuteromycetes or Basidiomycetes. Okay, so the correct answer is Deuteromycetes. Yes. So let's see the another question. Which of the following matches is correct here? You can see this is a match the following question, and let's match. So we have in one column 1, we have mucor, we have reproduction by conjugation, and we have Ascomycetes. Then next one we have the agaricus, and we have parasitic fungus, and we have Basidiomycetes. Okay, sorry, this is okay. Phytophthora, Aseptate, and Basidiomycetes, alternaria this. Okay, so the correct answer is alternaria is having sexual reproduction absent, and it is belonging to the Deuteromycetes group. Okay, so another one. Which one of the following fungi contains hallucinogens? So you have to look at it, you have to tell me the correct answer. Okay, Morchella esculenta, then we have Amanita muscaria, and we have Neurospora species, and we have Eustylago species. So I hope 5 seconds is over. This is a very simple and easy question, right? So the correct answer is Amanita. This is the toadstools. They are very, very important hallucinogens. Okay, so let's see the other one. Black rust of the wheat. Okay, so this is also called as stem rust. So black rust of the wheat is caused by which fungi? Alternaria, Eustylago, Puccinia graminis, or Xanthomonas oryzae. Okay, so I hope this is discussed also in the class. So the correct answer is Puccinia graminis triticea. This is the causing the black rust for the wheat. Okay, now, so which one is wrong in pairing for the disease and is caused and is causal organism? Okay, so black rust of wheat is caused by Puccinia. Next is loose smut of the wheat is caused by Eustylago. Next is root knot of the vegetables. This is caused by Melodogine species. And late blight of potato is caused by Alternaria solani. Which one? My dear students, I hope this is fine. So late blight of potato is not caused by Alternaria. Alternaria is the toadstools, right? So this is another one. So it's not, Alternaria is belonging to the Deuteromycetes group. This is not causing the late blight of the potato. Rest all are correct. Fine. Now, one more question. Which fungal disease spreads by the seeds and the flowers? So options are the loose smut of the wheat, corn smut, covered smut of barley, and soft rot of the potato. It's a previous year question. Okay, the answer is loose smut of wheat. This is caused spread by the seed and the flowers. Rest of them are not. Okay, so and after this, black rust of wheat is caused by Puccinia, Eustylago, Albugo, or Phytophthora. So this is already done once. So this one is caused by, definitely my dear students, it is caused by Puccinia. Okay? Okay. So now another question from the same type of topic, that is the black rust of the wheat. This is a fungal disease, right? So black rust of the wheat, this is a fungal disease, and this is caused by which of the following fungi? Albugo candida. Next is Puccinia graminis triticea. Next is Melamsora linea and Claviceps purpurea. So Which is the correct answer? I hope you got it. This is repeated twice or thrice. So black rust of wheat is caused by Puccinia graminis triticale. So very important fungi, my dear students. So please do remember this name. And so now we are at the end of the session, but this is not the end, my dear students. We can meet once more if you want. Okay, so once more, it will be really I will be very happy to meet you all in person, and to make you all my own students and to teach you properly, very nicely for the NEET botany subject. So in order to meet me and in order to take classes, coaching, everything from me and all these faculties which you are watching in the YouTube live sessions, you need to connect with us. Yes, so this is for your connection with us. You just make this connection. So how you can do this? Quickly scan this. You will be getting a registration form, and in that particular registration form, you will be knowing more about us, that is Knowledge Hub by Sri Chaitanya, and about our courses. So thank you, my dear students. Take care. Let's meet in the next session. Till that time,